Thanks everyone for coming. This is actually our third edition of running this little journal club. This comes off the back of the meeting in Crete in July, where we kind of started up this little stromal network. And Vasso and Christina are going to do our third iteration of the journal club. So yeah, I won't say any more for now. So take it away.
Great. So, hi everyone. Today we will be presenting this paper titled Cross-Tissue Human Fibroblast Atlas Reveals Myofibroblast Subtypes with Distinct Roles in Immune Modulation. You will first hear from Christina Paraskeva, who is a PhD student in Dr. Kollarakis' lab in Alexander Fleming. So I will just pass the mic on to her.
I would like to thank Vasso for introducing me and also for the opportunity to present this paper today together. As I said, I am a PhD candidate at Alexander Fleming in Athens. So today's journal club has to do with the paper entitled Cross-Tissue Human Fibroblast Atlas Revealed Myofibroblast Subtypes with Distinct Roles in Immunomodulation by Yang Gao, published this October in Cancer Cell. So to start with a brief introduction, what they're trying to do with this paper is to generate a human fibroblast atlas across different types of tissues, encompassing several different tissue conditions also, so disease states. And in order to do this, they actually integrated a large number of single-cell RNA sequencing transcriptomic datasets. So they do a comparative analysis of around 30 public datasets along with some newly generated transcriptomic data from their own lab. And as you can see, this covered a great number of human samples that encompassed a great variety of tissue types, including head and neck squamous cell, lung, liver, pancreas, kidney, stomach, colon, breast, ovary, synovium, and skin. And all of this also included different states actually of the tissues, including healthy tissues, inflamed, tumor, and metastatic tissues. With the term non-inflamed, they mean that the sample was acquired from a section adjacent to an inflamed tissue. And with the term normal, they mean that the sample was acquired from a section that's adjacent to tumor, but it's normal. And it's of great importance that they actually incorporate a great number of cells in this transcriptomic dataset, meaning that their dataset is greatly robust. And as you will see later on, they also validate their findings with in vitro stimulations in primary fibroblasts and with spatial transcriptomics. But Vasso will discuss about this. So let me note here that with the term inflammation, they refer both to the inflammatory condition and also in fibrosis, as they say in terms of simplicity. So with this integrative analysis, they managed to actually identify 20 different clusters of human fibroblasts that incorporated progenitor-like fibroblasts, myofibroblasts, inflammatory fibroblasts, some tissue-specific ones that were termed like this because they were enriched in some specific tissues like PRG4 positive that were found only in the synovium, fibroblast-like cells including smooth muscle cells, pericytes, and mesothelial cells, and some other fibroblast clusters that couldn't be categorized as a separate specific group. So in order to characterize all of these clusters, they actually calculated, let's say, the abundance of each one of these clusters in the different types of tissues. So they created frequency pie charts showing how these fibroblasts are distributed in the different tissues. And this allowed them to classify the fibroblasts in three major categories. The specialized ones, that are the tissue-specific fibroblasts found only in one or two tissues. The shared fibroblasts that were mainly enriched in one or two tissues, but they were found with lower abundance in other tissues as well. And last, the universal fibroblasts that were found across all tissues, or they were at least expressed in more than 8 tissue types at the same time. And going on, they did the same thing and calculated the abundance of fibroblast clusters now in the different states of the tissues. And what they observed was that the tissue-specific fibroblasts are enriched in healthy tissues or non-inflamed, meaning healthy-like state. The shared ones were distributed with a great variety in the different conditions, while the universal ones included the clusters that prevail during tumor and metastasis. Now here, we observed that there is a bit of, let's say, misunderstanding or something that is not going so well with the inflammatory fibroblasts. Because as you can see, the inflammatory clusters are actually more enriched in healthy or non-inflamed states rather than in the inflamed states. And let me say that this categorization of fibroblasts and the terminology has been given based on the enrichment of each cluster in different signature scores. So, for example, the myofibroblast clusters were enriched in myofibroblast signature genes, the progenitor clusters for progenitor signature genes, and the inflammatory ones for inflammatory signature genes. Now, let me note here that there were two clusters of myofibroblasts, the SFRP2 positive and HOPEX positive, that actually had a higher signature score for progenitor cells, for progenitor genes actually, while one of the two progenitor clusters, the PI16, appeared to have a higher score for inflammatory genes. Now, we tried really hard to find the genes that they use for these scorings, and they actually always prompt you with different references. And we were thinking as a comment that since they are trying to do a research paper, it would be interesting to incorporate these genes in a supplementary table or in their methodology section. So moving on, as we said, they observed that so they defined that these clusters were distinct in the transcriptome level with gene expression profiling, but they also tried to show if they are also regulated differentially. And for that reason, they conducted a cynic analysis that would allow them to see the transcription factors that control each one of these fibroblast identities. And as you can see, several clusters grouped together, meaning that they could be regulated by the same set of transcription factors. And they pinpoint the paper FOXP2 as a transcription factor that could drive the expression of progenitor genes, LEF1 as one of the transcription factors that strongly drives the myofibroblast lineage, and of course, as it is reasonable, NFKB1 that drives the inflammatory fibroblast lineage. And this is also very well depicted in the corresponding clusters in a human representation. So as I said, they observed that the tissue-specific fibroblasts are enriched in healthy state. And they wanted to see if these fibroblast communities are altered in the transition from chronic inflammation to fibrosis. So they decided first to depict the proportions of each fibroblast subset in the different conditions. And they observed that in the inflammation stage, you have a great retention of these tissue-specific fibroblasts with a slight increase for myofibroblasts, which is the purple one. Okay, while in tumor and metastasis, you have the highest enrichment for myofibroblasts and fibroblast-like cells. So they hypothesized that this inflammation stage is like an intermediate for the transition from the healthy to tumor phase. So they defined a scoring, an activation scoring for fibroblasts, and in the heat map you can see some of the genes that are used for this scoring, that, let's say, validates that this inflammation stage is an intermediate because it moderately expresses these genes, while in tumor and metastasis you have a high enrichment of this scoring. And this is also well depicted for several different types of tissues where inflammation poses as an intermediate state. But here we have the second inconsistency with inflammatory fibroblasts because, as you can see for the pink cluster, actually it's more abundant in healthy tissues, and actually there is the highest proportion there rather than in the inflamed tissues. So in the same context of trying to characterize how fibroblast, this community is altered in the transition from inflammation to cancer, they decided to focus first on the two clusters that have a great abundance in both states, inflammation and cancer. And these were the MMP1 positive myofibroblasts and the CD74 positive antigen-presenting fibroblasts. What they observed was that genes that are highly upregulated in the inflammation stage were actually pro-inflammatory cytokines that were correlated with immune-related processes, while in tumors, there was an enrichment for genes associated with the Wnt and TGF-beta signaling pathways, that the correlated pathways, the associated ones were TGF, Wnt signaling and also matrix organization and tissue remodeling for the cancer. And apart from focusing only on these two clusters, they moved on to also study the total fibroblasts, meaning the rest of the clusters. And they observed the same patterning occurring. In inflammation, you have an enrichment for pro-inflammatory and immune-related pathways, while in cancer, you have an enrichment for tissue remodeling, matrix metalloproteases, collagens, and again, Wnt and TGF-beta signaling. So these two states, inflammation and cancer, are different because different pathways are activated, but they are also distinct metabolically because they observed that during cancer, there's an enrichment for hypoxia and glycolysis, while there's a dampening of inflammation-associated metabolic pathways like retinol metabolism and DNA methylation. And later on, they have an illustration where they try to depict how the composition of fibroblasts changes in inflammation and cancer. So as you can see, some of the clusters actually appear in both states, like SFRP2 and MMP1 positive myofibroblasts, while others govern each one of the states, like LRRC15 positive myofibroblasts that govern the tumor states, and CD74 antigen present cells that govern the inflammation state. Now here we have some collective comments regarding the inflammatory fibroblasts because someone ends up asking himself which are the inflammatory fibroblasts. At the beginning, it seems like it's the IL-6, HGF, and HSPA6 positive cells, but these actually seem to be more enriched in healthy tissues, as we said, rather than the inflamed ones. And later on, they give you the hint that SFRP2 and MMP1 have inflammatory cues, but they also have myofibroblastic cues. And then this is a little bit, let's say, it confuses you. It becomes confusing. Later on, they will focus on the tumor part. And since they observed that the myofibroblasts are the ones governing the tumor states, they focus on these subsets. And these were the LRRC15 positive, HOPEX positive, MMP1 positive, and SFRP2 positive fibroblasts. As common characteristic, they highly express all the myofibroblast signature genes, but they also have some differences in transcriptome level since they express different genes and they are enriched in different pathways. For example, as you can see, SFRP2 and MMP1 have an enrichment for some inflammatory genes like SFRP1 or HIF1A or 6L1, while LRRC15 and HOPEX are more towards the myofibroblast side with MMPs, HOPEX, and collagens. So they conducted a trajectory analysis with several types of bioinformatic tools where they managed to show that LRRC15 fibroblasts are the more terminally differentiated one. They are at the edge of the trajectory. And they can actually originate either for progenitor fibroblasts, the cluster C5 and C3 of PI16 and collagen 15 fibroblasts, or from pericytes, cluster 8. And they, regarding the pericyte origin, they try to validate this by using some immunostainings where they saw that the LRRC15 positive myofibroblasts actually express some pericyte markers like NCAM or PDGFR-beta. We have the question marks here because we think that it's very, very difficult to actually prove colocalization with this type of stainings because pericytes with myofibroblasts are in a very close proximity. So we consider that more experiments would be necessary here to actually prove that pericytes are an origin for myofibroblasts. We will discuss this later on. So again, regarding the trajectory analysis, okay, there are some other myofibroblast clusters, as you can see, like C16, C19, and C6 that they were thought to be originating from the progenitor-like clusters, and C17, C9, C11 that they mainly originate from the pericytes. Okay, so these were considered intermediate states that originate from different sources. And the first thing that we asked ourselves were which tissues and which states did they use in this trajectory? The healthy ones, the tumor one, which ones? Because if you see, for example, they make an effort to prove this, but in skin, for example, you have a very small number for myofibroblasts, while in pancreas, myofibroblasts are enriched. So the one seems to be healthy, the other seems to be a tumor. So we would like to have the information of the tissue state for this trajectory analysis. Moving on. Okay, regarding the origin of parasites, as we said, more experiments would be needed. Either murine models to prove with a lineage tracing that parasites can give myofibroblasts, or some in vitro experiments. For example, since they have the c-Myc, they could overexpress the transcription factor that governs myofibroblasts in parasites to prove that parasites can convert to these LRRC15 myofibroblasts. Now, to sum up with this, we said about the intermediate transitional states of myofibroblasts and the terminally differentiated LRRC15, so we covered this. And we have to say that these are different because they are enriched in different pathways. So the LRRC15 terminally differentiated are more enriched for TGF, Wnt, and PI3K. The HOPEX seem to be closely correlated with LRRC15. This is also obvious from the trajectory. You have high TGF and Wnt in this as well. But SFRP2 and MMP1 seem to be a little bit more inflammatory as well. You have an enrichment for TNF, NF-kappa B, and hypoxia, mostly for the MMP1 positive myofibroblasts. And they also try to give a translational, let's say, outcome from all this analysis. So they managed to show by the proportions and the hazard ratio in patients that patients with highest proportions of LRRC15 myofibroblasts actually have the worst outcome and poorest survival. And last but not least, they also, to further, let's say, depict the differences of these fibroblasts, they conducted an ISNET analysis to show the external stimuli that could drive these lineages. So TGF-beta was a stimulator that could drive all of these myofibroblast identities. But for MMP1 and SFRP2, other potent stimuli were more inflammatory-like, including interferons, TNF, and IL-1 beta, while for HOPEX and LRRC15 that were most closely correlated, SPP1 also seems like a potent stimulator. So for the two intermediate states, we have some pro-inflammatory like stimulants also and a pro-inflammatory program. So these two clusters seem to be more inflammatory-like, while HOPEX and LRRC15 seem to be more pro-fibrotic-like because SPP1 is also a marker, let's say, or gene closely correlated with fibrosis. And I will pass on to Vasso now.
I hope you can still see my screen here. Right. So can you hear me, first of all? I hope so. So next up, they focus on the two progenitor-like states. Let me see if I can hide these. So they focus on the two progenitor states, PI16 positive and collagen 15A1 positive fibroblasts that can both differentiate into myofibroblasts. But despite both having a progenitor signature, they exhibit differences in gene expression as seen here. So PI16 positive fibroblasts upregulated genes related to TNF signaling, hypoxia, and vascular remodeling, whereas collagen 15A1 positive fibroblasts upregulated genes related to adipogenesis, basement membrane, and regeneration. And interestingly, they find that the PI16 progenitors specifically were enriched in non-cancerous adjacent to tumor regions compared to LRRC15 terminally differentiated myofibroblasts that were most enriched within the tumor itself. So given that, and the niche-net that they conducted for the two populations in tumor, they find that PI16 positive cells are governed by inflammatory predicted stimulants, whereas LRRC15, as Christina mentioned, is governed by TGF-beta signaling. And they said, wait, this kind of looks like the ICAPs and MyCAPs that we know and love from PDAC, right? So they wanted to see whether or not PI16 positive progenitors kind of look like the ICAPs from PDAC. And to do that, they performed some in vitro stimulation by getting the fibroblasts from adjacent normal tissue from lung tumors and colon tumors, and stimulated them with LEAF, CXCL12, sorry, IL-6, and they got a progenitor-like signature, like the one depicted here. These are some of the top genes in this expression signature. But when they stimulated them with IL-1 and TNF-alpha, they got an ICAF-like signature, like the one from PDAC. So they might be both regulated by pro-inflammatory programs, but they're different from ICAFs that we know. And to further validate that, they mapped these LRRC15 positive population and the PI16 positive population on tumors from spatial transcriptomic data that they got from other studies and produced in-house. And they saw that LRRC15 positive population is enriched in tumors and right next to tumors, whereas the opposite is true for PI16 positive fibroblasts. And also when they mapped the stimulation signatures that they got from their in vitro stimulations, they also observed that stimulations leading to progenitor-like expression were distal to tumors as well. So next up, they tried to stratify patients according to their fibroblast cluster proportions, and they obtained 4 different subgroups. The cancer type specific group, which is here, a mixed group that had a mix of fibroblast populations within it, but most samples actually fell into one of 2 categories: the myofibroblast high, progenitor fibroblast low subgroup, or the opposite, the myofibroblast low, progenitor high subgroup. What is interesting here is that they found out that the higher your proportion for terminally differentiated myofibroblast, the worse your prognosis and outcome is. So this was quite important for the translational benefit of this classification. Some thoughts we had on this, though, was that the sheer abundance of breast cancer samples in these stratification might drive even the classification towards a very specific pattern. And we also wanted to note that people that work with, gut, we wanted to note that the GI tract cancers, that the tumors that are all classified in the tissue, in the cancer-specific subgroup, often form as adenomas and then progress into carcinomas. So we would like to see some actual association with tumor staging within this, and whether or not as the tumors progress, we have an accumulation of these LRRC15 positive myofibroblasts. So then moving on from their spatial transcriptomic data, they wanted to infer multicellular modules that might be at play here. So they assessed the co-occurrence of fibroblasts, T cells, and myeloid cells in their spatial transcriptomic data. And they actually really nicely demonstrated that within these multicellular modules, the PI16 positive progenitor-like cells segregate completely from the LRRC15 positive terminally differentiated myofibroblasts. The PI16 positive are within the CM1 module, and the LRRC15 positive are within the CM6 module. They also show that the PI16 positive progenitors colocalize with effector T cells, whereas the LRRC15 terminal myofibroblasts colocalize with SSP1 positive macrophages, contributing to an immunosuppressive and pro-tumor signature. And they also find, and they note here, that the CM7, the multicellular module 7, includes MMP1 positive myofibroblasts that might have a more inflammatory profile that Christina mentioned, but they colocalize with Tregs and immunosuppressive DCs in the tumor microenvironment, so they're considered also pro-tumorigenic. So basically, what I said before, when looking at patients that have a high proportion of CM1, CM6, or CM7, they find that the ones that have higher proportion of CM1 modules are enriched in signatures of effector cells, NK cells, and anti-tumor cytokines, where the opposite is true for the patients that are enriched in the CM6 module. So one is anti-tumor, the other one is pro-tumor. And we should note that even though they focus on that, another program contributing to anti-tumor immunity is the CM4, which includes the antigen-presenting fibroblasts that Christina mentioned in the beginning. And one contributing to pro-tumor immunity is the CM7 that I mentioned, which includes the MMP1 fibroblasts that contribute to immunosuppression via communication with Tregs and DCs. So then they focus a little bit on the LRRC15 positive fibroblasts and SSP1 macrophages within the CM6, and they show from the spatial data that these are actually very close together, these two cell types, which they also confirm through immunohistochemistry. The asterisks are LRRC15 positive fibroblasts, and in the dashed circles are the SSP1 macrophages, which they're pretty close together here. But because LRRC15 fibroblasts are not the only ones within this specific module, they asked whether this colocalization is specific for them and not other fibroblast types. So they performed an analysis where they compare FAP positive LRRC15 negative and FAP positive LRRC15 positive fibroblasts and their closeness to SSP1 macrophages, and they find actually that the latter are relatively closer to SSP1 macrophages in their data. So lastly, they wanted to predict interactions and ligand receptor interactions within the different multicellular modules. So they perform a cell phone analysis for ligands and receptors within each module. And here we have the pro-tumor modules. We have a chord plot for the CM6, where we have the terminal differentiated fibroblasts and macrophages expressing SSP1. So they predict that one of the ligand pairs that is most significant for this is integrins with SSP1, ADAM12 with integrin B1. So anyway, matrix remodeling and matrix remodeling promoting interactions, which they also map here. And for the CM7, we have a chord plot predicting interactions between MMP1 positive fibroblast Tregs and immunosuppressive DCs. They also plot some of these ligand receptor co-occurrence in their slides, and they conclude that some of the signatures that these ligand receptor interactions contribute to are matrix remodeling, of course, hypoxia, and IL-6 JAK-STAT3 signaling that collectively are pro-tumor. And for the other side, the anti-tumor predicted interactions from cell phone, we only have a chord plot for the CM1, but basically we can see that the PI16 positive progenitor-like fibroblasts communicate with effector CD8 cells and LYV1 macrophages. One of the top, let's say, interactions was actually that of CXCL12 12 and ACKR3 and CCL5 with ACKR4 that contributes to driving the infiltration of these tumors with effector cells. And basically, we can see that by the most prevalent signatures that they also mapped on these spatial transcriptomic slides. And the second anti-tumor immunity promoting module is that of CM4, which includes antigen presenting CAFs, which through CCL19 and CCL21 promote the infiltration of T cells and B cells within the tumors and contribute to anti-tumor effects. So after all that, this is a table that summarizes all of the characteristics of myofibroblast clusters, what might drive their activation, what they contribute to, which is bad and which is good. We can take a look at it while we discuss after. And basically, they provide a summary in their last figure of the fibroblast cell features, dynamics, and spatial niches in this study. So I copied this directly so we don't, so I don't paraphrase it or misunderstand anything. So basically, they propose that we have PI16 positive fibroblasts primarily found in adjacent normal tissues that can transition into SFRP2 fibroblasts, and when they are exposed to IL-1 beta and TGF-beta, and that these specific fibroblasts are key to attracting memory T cells to perivascular niches mediated by this interaction. Then we have the collagen 15A1 positive fibroblasts that are predominantly found in adjacent normal tissues and close and near basement membranes that can be transformed into the other intermediate fibroblasts, the MMP1 positive ones, when exposed to interferon and TGF-beta stimulation. And these are key in producing an immunosuppressive environment by attracting Tregs and myeloid cells like LAMP3 positive DCs. And that basically MMP1 positive, SFRP2 positive, and this forgotten HOPX positive myofibroblast that derives from pericytes can all give rise to LRRC15 terminally differentiated myofibroblasts under the influence of the master regulator of myofibroblast TGF-beta. And finally, that these LRRC15 positive fibroblasts, through attracting SSP1 positive macrophages, contribute to a loop of immunosuppression that drives immune cell excluded tumors. So some final thoughts from Christina and I. We did this together. We really liked how novel this study was, since it provided a great resource for a first cross-tissue human fibroblast atlas of its size. We upload the amount of both single cell and spatial transcriptomic data provided and generated newly for this study. We also liked how it provides some food for thought, especially this trajectory analysis that we're kind of skeptical about, but could be the basis of in vitro validations. And we believe that it would be worth it to compare them to other mouse atlas, similar to these. We also like the translational benefit of this because stratifying patients based on a more complete view of the tumor microenvironment can actually be very beneficial. And overall, we liked the approach because we like seeing inflammation and cancer as a continuum. So we really appreciated that in the first half of the paper. And for some considerations, as Christina mentioned, we're a little bit iffy on what exactly the identity of inflammatory fibroblast in the original subtyping was. And especially Christina, who's working on this, we would like to have seen some underrepresented activated conditions like regeneration and wound healing. Of course, this is hard to do in human, but you know. And our major issue were the trajectory analysis. We would have liked to have seen some more validation experiments of those, especially the PI16 fibroblasts were a bit, had us scratching our heads a little bit since they were actually quite inflammatory in nature, in contrast to other reports of this population. And as we mentioned before, on the translational benefit side, we would have liked to have seen some staging of the different tumor types. And last but not least, we need to have some nomenclature unification across studies. So, for example, even though they mentioned the PDAC study of MyCAFs and ICAFs, it was not clear the connection between known CAF subtypes and the myofibroblasts in their tumor studies. So with that, we're done and we're open to discuss.
Thanks. That was really nice, Christina Vasiliki. You've condensed quite a huge paper actually with all the data and made it very clear. And so we have like 10 minutes here, like you said, for discussion before wrapping up. Does anyone have any questions to start off? Or otherwise, I had a few thoughts as well. Yeah, I guess, yeah, definitely agree with your part about like kind of the trajectory analysis. I think the relationship between fibroblasts as they transition through these states is quite important. Especially, yeah, you mentioned about the underrepresented activation conditions, because I wonder with these kind of fibroblasts becoming terminally differentiated, is it because of the chronic environment, or is it really an early event that says, okay, these cells are transitioning to become the LLC? If you have that kind of, maybe if you keep up that last diagram would be kind of helpful. The on slide 25, Vassos. Yeah, so you see how they have this kind of arrows pointing towards the LRC15. What do you think? Do you think that it's really like the chronic stimulation of these fibroblasts, or rather a kind of initial event that says, okay, now these are going to become the LRC15?
Vasu, do you want to answer or do you want me to answer this? I mean, have the discussion about it.
No, go.
Okay, so in my point of view, even if you see the proportions, when they categorize the patients in different categories based on the tumor microenvironment, if LRC15 were the terminally differentiated, why are they not governed by this? I mean, you have terminal stage tumors, so why don't you have a tumor type that is governed only by LRC15 myofibroblasts? I mean, the relations are so close, and we all know the trajectory analysis. If you have tried to do it, it's actually a bit, how can I say it? Just a prediction that it doesn't always take into account the chronic series of events. Let's say this continuum, it's something that you have to predispose the program so that it will give you a result, especially when we're talking about fibroblasts. If you had stem cells and terminally differentiated epithelial cells, it would be more accurate, let's say, in a healthy condition. But for fibroblasts and across all conditions, it will show what it wants. You understand what I want to mean.
Yeah.
PI16 that were more inflammatory are actually deriving from collagen 15, which actually the opposite of what Bachler has said in his own paper with murine mice, with mice. So
with the in vitro stuff, did they like wash out the stimulation and see if they kind of go back as well? Because I guess I'm trying to understand if it really is like a one-way street with these kind of progression. Like you said, I know you have to kind of interpret the RNA velocity, but I wonder if it is like you disrupt the homeostasis of these fibroblasts and they're really going to stay like that forever, or they can kind of come back to a normal environment.
So you mean if they are permanently activated, or if they could revert and go back to their original state?
Yeah.
My personal opinion is that for some clusters, no, but still it needs a lot of validation to be done. And for humans where you don't have the regeneration phase to show a normal-like transition, because sampling and from these types of conditions is very, very hard in humans, you cannot say definitely. I mean, since it is my fibroblast, and if it highly expresses genes like alpha SMA, FAP, or some collagen, if it towards the CAF, let's say, state, it will be permanently activated and not be able to revert back. But they have some clusters, I think C6, C16, the lowest in the trajectory, the inflammatory, let's say, more like C16, C6, they're very close, C17, they're very close to the progenitors. These maybe, if they are not terminally activated, could revert back.
Cool.
They should have taken these cells with sorting and pour those stimulants on top to see if they will become progenitor cells. This would be interesting.
Yeah. And also just because now you're on this graph, like the parasites are really, they do seem really distinct. So I'm really curious about how they are really biologically related, how they can generate these RC15 cells.
It's like they had to incorporate it in the trajectory analysis. I mean, it's the only cluster that it's not a myofibroblast, and they didn't include all the other clusters of fibroblasts as well. It's like we just put the parasites in this trajectory because it was able to show an origin. Okay.
Yeah, I wanted to thanks by the way. It's really cool paper and really well presented by you too. I was wondering here, but actually with the trajectory, I don't know what you think about it, because they basically pulled all the fibroblasts they found from different organ systems, right? And anyway, they do this actually throughout their whole analysis, but they kind of remove in this way if there might be potential like organ specific populations or anything, right? I mean, in the end, they kind of split it again, but what do you think about this? Could they have done this differently?
You mean the trajectory analysis or the
Or
tissue?
generally, actually, right? Because they basically put everything on one big pool, which is in a way cool, but also like how much is driven by organ specific things.
Okay.
I mean,
Vasso.
yeah, if I may, just, deriving calves from parasites, right? They didn't reinvent the wheel. There have been reports of that, right? But in these reports, it is actually tissue specific. Tissues enriched in, blood vessel-like structures and stuff, you might get your calves from parasites, right? So this is one of the things that we observed when they broke down some of their trajectory analysis to specific tissues. So completely, I agree with you that that might be tissue specific. And if we go back to the stratification data, so the proportion of the different fibroblasts in different tumor types from different, actually cancer types from different tissues, you can see that in some, so for example, this whole column is just GI tract cancers. It has nothing to do with the LRC15 high, set that they have here. And I don't know if this is the staging of the tumor or is it because it is actually a tissue-specific effect that they see here. So I think it would be worth it to, apart from pulling everything together and creating this atlas, it would be best to also look into tissue per tissue to find these effects. I don't know, Christina, did you like to comment on something more on this?
Yeah, usually it's difficult when you have different states actually. So if you're trying to compare healthy tissue with a tumor tissue, and we're talking about fibroblasts, we're not talking about epithelial cells where you have, for example, stem cells, and it's very easy to show a trajectory. So if you try to compare fibroblasts across different states, it's just a prediction. It may show you something that truly happens, but it can also be arbitrary. For example, for me, if I compare healthy, inflammation, and regeneration, because regeneration, it's more normal-like, it will give me a trajectory that it's like healthy, regeneration, inflammation. And actually inflammation occurs before regeneration. So it has to do, I think, with the staging, with the chronicle order of facts. So if they have pulled everything together, they may have overestimated something. In my point of view, I will group them based on the state and per tissue, but first on the state, like all the healthy samples together to see what's happening.
Yeah, I think I like that
I
interpretation
like
as
that
well.
also. I like that also. I mean, not to, beat a dead horse, but these PI60 positive fibroblasts were super activated in this one, not like other, studies of this, of this kind anyway.
Great. Well, that was a super interesting discussion. I think we will just wrap it up now for the purpose of time because everyone's joining from different time zones, I guess. But thanks again, Christina and Vasilika. That was really, really excellent.